This protocol describes a method to reprogram blood mononuclear cells to neural stem cells that can be used to treat neurodegenerative diseases such as Parkinson's disease. It offers an autologous cell source to treat neurodegenerative diseases. Also, the time frame required for conversion to induced neural stem cells and differentiation to desired cell types is shorter than for induced pluripotent stem cells. Induced neural stem cells possess a good proliferative capacity and can be scaled up before application.
Since the iNSCs can be specified into different region-specific neuron cells, such as spinal cord neurons or motor neurons, this method may be extended for the treatment of other neurological diseases. The whole procedure takes a long time, with all steps related and critical. The visual demonstration gives an idea about how the cells look like and provides some tips to ensure a successful generation of the desired cells.
To begin, transfer previously isolated peripheral venous blood into a 50 ml conical tube and dilute it with an equal volume of sterile Dobecco's PBS. In another 50 ml conical tube, prepare 15 ml of sterilized density gradient medium. Tilt the tube at a 45-degree angle and slowly and carefully lay 30 ml of diluted peripheral.